Abscisic acid, ABA, is a crucial plant hormone that regulates various physiological processes, including seed dormancy, stomatal closure, and responses to environmental stresses such as drought and salinity. The effectiveness of ABA in mediating these responses depends on its precise transport within plant tissues and its perception by specific cellular receptors. ABA transport occurs both at the cellular and long-distance levels. At the cellular level, ABA can move via passive diffusion across membranes due to its weakly acidic nature, but this process is limited by the pH gradient between the apoplast and cytosol. More importantly, active transport mechanisms play a significant role in controlling ABA distribution. Several families of membrane transporters have been identified as key mediators of ABA movement, including ATP-binding cassette, ABC transporters, and nitrate transporter 1 peptide transporter family, NRTN-PTR or NPF proteins. For example, ABCG25 functions as an ABA exporter, releasing ABA from vascular tissues into the apoplast, while ABCG40 acts as an importer, facilitating ABA uptake into guard cells. NPF transporters, such as NPF46, also contribute to ABA import into specific cell types. These transporters enable plants to finely regulate ABA concentrations in different tissues and cells, allowing for rapid and targeted responses to environmental cues. The perception of ABA is primarily mediated by a family of soluble intracellular receptors known as PYR, PYL, RCR, pyrobactin resistance, PYR1-like, regulatory component of ABA receptor proteins. Upon binding ABA, these receptors undergo a conformational change that enables them to interact with and inhibit type 2C protein phosphatase, PP2Cs, which are negative regulators of ABA signaling. Inhibition of PP2Cs releases SNF1-related protein kinases 2, SNRK2s, from suppression, allowing them to phosphorylate downstream targets that drive ABA-responsive gene expression and physiological responses. This core signaling pathway operates in various cell types, including guard cells, where it mediates stomatal closure during water deficit. Overall, the mechanisms of ABA transport and perception involve a coordinated network of transporters and receptor-mediated signaling. The spatial and temporal regulation of ABA movement, coupled with precise hormone sensing, ensures that plants can adapt effectively to changing environmental conditions.